Hi, everyone. I'm Uri Manor. Our next speaker will be Dr. Ka Shu. Ka, you should try to share your slides now
Oh, all right. Yeah.
so we can get up and running. Go ahead and start that process while I introduce you. For those of you who don't know, Ka is a professor of chemistry at UC Berkeley. He has won numerous awards, including the Pew, Sloan, Beckman, NSF Career. He's a Chan Zuckerberg Biohub investigator, and he recently won the, well, not recently anymore, the DP2 Innovator Award. And Ka has been doing some really interesting work taking advantage of some of the properties of the camera hardware to improve super resolution microscopy. So this is a really interesting area of exploration and development that I hope we will hear a little bit about now. I
All
see the
right. Can you
slides
hear
are
me?
up. All
Yeah.
right, take it away.
Okay,
Thank you.
great. Thank you. Thank you very much for the invitation. Today I'll be talking about adding additional dimensions to super resolution microscopy, as alluded actually by one person actually asking the question, we can get more information beyond the position of molecule. So, well, I think AK gave a very nice introduction that by looking at the blinking of molecule, we can collect the position of molecule and through that generate a high resolution image, gradually like showing in here for this microtubule. That has been around for more than 15 years or almost 20 years. But what I'm going to talk about is that indeed we could encode and decode different dimensional information into that, and hopefully by combining all those different frames of data, we should be able to generate a high resolution image containing those different information. And I'll be mostly talking about two examples of the spectrum of single molecule and the motion and diffusion of the single molecule. For spectrum, here we are looking at the single molecule images of a camera, but we showed if we just insert a prism into the light path, we can directly disperse all the single molecule fluorescence into the spectrum. So they are, as you say, they are dispersed, elongated in the horizontal direction. Those are the single molecule spectrum. So now if we have the single molecule blinking, where we typically do to collect the positions of molecule. Now we have the blinking spectrum of all those molecules. So all those stripes are actually individual molecules showing their emission spectrum. So now we can collect thousands of single molecule spectrum in just 1 second, and in 5 minutes we have millions of spectrum. And we also have the position of the same molecule that enable us to do spectrally resolved single molecule localization microscopy. Um, we apply this method to imaging the membrane in the cell using a dye called NELRAD. This dye emits different colors in different chemical polarity environment, and we showed by detecting the position of each molecule, for each detect molecule, we plot a position as a dot, but we can also color it based on the emission wavelength of that single molecule. Then we generate this spectrally resolved SMLM image using this dye, and we immediately see different colors showing up in the cell, so that the plasma membrane enclosing the cell is blue in color, but the internal membrane of organelles are yellow in color. We can further just look at the average single molecule spectra for those different parts of the cell, and we can see essentially the internal membrane have this redder emission spectra, but the plasma membrane have this bluer emission spectrum. So that shows by having both high spatial resolution and also the emission spectrum of each molecule, we can now reveal different contrasting properties of the membrane to say that the plasma membrane in this case is more hydrophobic and the internal membrane is more hydrophilic. So essentially that's what we were able to show in this case by having spectrally resolved super resolution microscopy. We also demonstrated another direction to look at single molecule velocity and diffusivity, which has been hard to look at for the regular imaging camera frames with 10 millisecond frame. There are a lot of motion blurs of single molecule. We developed an interesting trick where we have the two pulses across a pair of frames, so we can try to measure the motion of molecule within those short time window determined by the pulse sequence. And if we repeat it a lot of times, we can do statistics of those molecules. So that essentially look like this. We have a lot of pulsed frames, and we can map the displacement within 1 millisecond for many molecules and generate a histogram to find out local displacement. But we can also use a spatial bin to bin down. In each spatial bin, we have maybe hundreds of molecules. We have a displacement. We can have a local diffusion rate associated with that mapping. And then we can generate a colored map, giving us a local diffusion at the super resolution level. For example, we showed inside the nucleus, there are local inhomogeneity in the diffusion rate for a general fluorescence protein. And if we compare those patterns with the local staining of the DNA, we show the fast diffusion part of the cell of the nucleus is the region where it is not occupied by the chromosome. But for region with high density of chromosome, with high density of DNA, we see a very slow diffusion. So if we overlay those two results from different dyes, one is labeling the DNA, one is just free protein diffusing, we see they overlaid really well so that all the fast diffusion are empty space, but the slow diffusion are essentially dominated by the chromatin structure. So that tells us the chromatin ultrastructure can modulate the local diffusion in the nucleus. I want to end up with a recent development that we did to enable really fast diffusion rate tracking at microsecond time resolution. This is based on the EMCCD working principle that it had a very fast way to vertically shift the image at below 1 microsecond per row. So what we did in here is that after each exposure of about 100 microseconds, we just allow the image to shift by 10 rows within a few microseconds. And we can repeat this process a lot of times so that within one frame of the camera, we have a lot of those shifting going on. And in the end, we generate a streak of image on this camera frame so that eventually we can then repurpose just to flip those image back to generate the original trace of each molecule. So the raw data look like this. We'll have a lot of streaks essentially recorded on the camera, but we can just reconvert it back to generate a trace of single molecule, and we can use them to map the very fast diffusion of single molecule. Actually, we can do it now down to 60 microsecond time resolution, and based on that, we actually can get very fast diffusion results. We were able to also use it to eventually to do mapping inside the cell to give us very nice mapping results of the fast diffusion inside the cell. So together, I hope I've been able to show with a few examples that we can go beyond the localization of single molecule to give us additional functional inputs, especially like spectrum and motion of the molecule. But the final goal is we want to go beyond the structures to give us new information inside the live cell and also in other environment, so we can look at those parameters inside the live cell. So if you are interested in those topics, we have a few review articles, but also account in chemical research, essentially discussing those type of very interesting discussion going beyond the imaging aspect of super resolution microscopy. So with that, I'd like to thank my lab and also funding agencies, and I'd be happy to take more questions maybe in the discussion session because we're running out of time, I guess. All right, thank you very much.